Time. Okay, so my lab really does not study evolution per se, except in the way that we all study evolution implicitly. If you were at the gene regulation sessions yesterday, you heard a couple talks about neural crest development, which is a very important tissue and vertebrate development relevant to evolution. So we've been interested in for a long time in how the neural crest produces the varied derivatives that it does. And in particular, we've focused on pigment cell development, the process is in going from a multipotent neural crest cell to a differentiated pigment cell. And so as we heard alluded to yesterday, decades of work in embryological and genetic and molecular experiments in mouse and chick and amphibian systems have led to the prevailing view that this is by a progressive fate restriction that occurs in neural crest. So in zebrafish and in most cold-blooded vertebrates, we have the added complexity or the added kind of interesting feature that the pigment cell, we have not just the single type of pigment cell in these organisms, but actually multiple other types of neural crest-derived pigment cells. In zebrafish, the yellow xanthophores and the shiny silvery iridophores. And of course, the studies from mouse can't really tell us a whole lot about the processes that produce these pigment cells. So one gene that my lab has really focused on over the years, which actually did originally arise from mouse, is a classic mouse mutant, MITF. So this is the microphthalmia-associated transcription factor. There are upwards of 20-odd alleles of this mutant that was first identified in the 1940s, and this just shows one of the alleles here. So this gene, as alluded to yesterday, is a master regulator of melanocyte development. So if you have mutations in this gene, you basically completely lose the melanocyte lineage, but other neural crest lineages are not affected. So this MITF encodes a basic HLH leucine zipper transcription factor, which binds to DNA as an obligate dimer to regulate gene expression. So I've been working on this gene for a long time, and I try to keep up with the literature as well as I can. But it was actually just a matter of months ago that I learned I was living with a potential MITF mutant, and that the variations in the MITF locus are actually thought to be responsible for a lot of the what's called the extreme white spotting phenotype in dogs and many other domesticated animals. So there you go. So a long time ago when I started working in zebrafish, I started working on this mutant here, which mutant completely lacking neural crest-derived melanophores, and this turned out to be a mutation in one of the MITF orthologs of the zebrafish. So completely missing melanophores, but and so many of you are probably familiar with this line from the CRISPR strain or other using it for other sorts of experiments. But you may not be aware that these, in addition to missing melanophores, these fish actually also have an increase in the number of cells in the iridophore lineage. And if I may make just one other aside, I noticed from the talks yesterday that the artwork from the Philo game card deck that everyone was given, if you look at the zebrafish card, if I'm not mistaken, I think that could be a MITF mutant adult zebrafish that's on the card. I need to consult with the artist to find that out for sure. But okay, so with the MITF mutant, then we see a loss of the melanophore lineage, but we see an increase in the iridophore lineage. And we haven't nailed this down absolutely, but we think at least part of this may be, we think there may be an actual fate switch involved in this. Okay, so along with MITF, there are actually a few other genes in the same gene family of these transcription factors. In mammals, there 3 other genes. In zebrafish, due to duplications, we have a total of 6 genes in what's called the MIT family. And so we were interested in, in mammals, none of these genes appear to have a role to play in neural crest development, but we were interested in the zebrafish, whether any of these genes might have such a function. And looking at so we cloned these out and looked at the expression of these, and a gene that was introduced in a talk yesterday, tfec is the one other gene in the family that has neural crest expression, and it's actually fairly interesting that it's actually expressed somewhat earlier than the MITF gene, and you can see here that, so comparing embryos at the 20 somite stage, the tfec expression is actually fairly down most of the extent of the axis, whereas at the equivalent stage, MITF is still fairly anteriorly restricted and is about where tfec was expressed at the 16 somite stage. All right, so we had this, and I want to point out as well, which Cleo showed in her talk yesterday, but this early broad neural crest expression later becomes restricted to cells that are basically where you expect to see pigment cells of the iridophore lineage, so in the dorsal and ventral edges of the trunk and tail, and in these patches over the yolk, and in cells covering the surface of the eye. So, of course, we wanted to know what function this gene might potentially play in iridophores and other cells. So we started off making morpholinos, eventually found a couple nice splice-blocking morpholinos that worked, and gave us this phenotype that we were hoping for, in that we get animals growing up that are basically missing most, if not all, of their iridophore. So you see over the eye, the eyes are much duller, and you're missing these big patches of iridophores that you usually see over the yolk. So that was promising, but of course, we really wanted to generate a true mutant in the locus. So we started off, we made a couple of different TALENs, and then TALEN pairs, and then found a CRISPR guide RNA that worked well, and so I'm just going to tell you today about the results with the CRISPR, so targeting basically this HLH leucine zipper domain encoding exon that's absolutely important vital for the function of this gene. So, so this is that we got this nice guide RNA where we injected into embryos, and we can actually see a phenotype when we grow up the injected fish, and that we're actually missing. We saw that we were missing iridophores in the embryos, but we grew these fish up, and we see a continued loss of iridophore pigmentation here. You can see in the eye and in the body. So then we were able to, from these, identify several germline transmitting founders, and we're happy to see that so this is a couple of an F1 embryo that we continue to see the same phenotype of loss of iridophores over the eye and the patches over the yolk. Okay, so then it looked like sort of maybe in the same way that the MITFA protein is required for the melanophore lineage, that this tfec BHLH leucine zipper protein is required to produce iridophores. Now, of course, we wanted to know what a double mutant looked like, and we weren't so surprised initially to see that the double mutant, is lacking both melanophores and iridophores, but what did sort of surprise us was, it's a little difficult to see here, but we're actually now missing also the third neural crest pigment cell type, the xanthophores. Actually, this wasn't entirely a surprise because we'd seen this with when we injected the tfec morpholinos into MITFA mutants as well. But it was nice to see it was from the mutant that this wasn't just some sort of morpholino artifact. And so it's easier to see the xanthophores here, where we've incubated the embryos in methylene blue, which people use to retard fungal growth, and they're with their embryos. But the xanthophores, you may have noticed if you use this, that the xanthophores will actually take up the methylene blue and become stained this color. So in the TFEC single mutant, there really isn't much of an effect, if any, on the xanthophore pigmentation. But here you see with the double mutant, we basically don't see any blue cells at all. You can see that this embryo was at least sitting in some methylene blue because it maybe swallowed some water that had the blue in it. So if we look, and likewise, if we look at a early marker of the xanthophore lineage, xanthine dehydrogenase, there is maybe a slight reduction in expression in the TFE single mutant, but there are hardly any expressing cells left in the TFEC MITF double mutant. Okay, so then it looks like the combination of MITFA and TFEC mutants eliminates actually all three of these pigment cell lineages. I don't have time to show you our data we've gotten so far that suggests that the other neural crest lineages, cartilage, and as well as neural and glial derivatives are not affected. But so the MITF TFEC double mutant kind of superficially resembles the colorless mutant that you may be familiar with, mutation in SOX10, where you lose all the pigment cell types, hence the name. But not so easy to see in colorless is that you actually have profound reductions in the other, basically all the neural crest cell types except for the jaw cartilages. There's another mutant called Shady from the original screens 20 years ago, encoded by the LTK receptor tyrosine kinase. And so this mutation, so this is another gene that affects actually only the iridophore lineage. And so we were interested in the relationship of TFEC and MITFA and these other genes. So we've just started to look at this, and we were wondering if TFEC would be, is it going to be regulated by, like MITFA is, or like LTK. So LTK is actually still, even though colorless SOX10 mutants lack iridophores, they still retain expression of the LTK receptor tyrosine kinase. However, SOX10 is absolutely required to get MITF expression in the melanophore lineage. So it turns out that TFEC expression is actually still retained in colorless SOX10 mutants. It may be slightly reduced, but this suggests that maybe TFEC is regulated in a similar way to LTK rather than MITFA. And then lastly, we've looked at LTK expression in the TFEC mutant. So as I just showed you and is shown here, you still have LTK expression in the SOX10 colorless mutant, but it appears in the TFEC mutant that you lose LTK expression. So the model we have here at the end is actually, there isn't much of a model here. There are a lot of gaps, basically, which will hopefully be filled in by the kind of GRN studies that you heard about yesterday from Robert Kelsch's lab. And further experiments that we'll do. But so SOX10, while it's required for MITFA expression, does not appear to be required, at least. It may still have some role in regulating these other genes in the iridophore lineage. But this suggests that the lack of xanthophores and iridophores in the SOX10 colorless mutant may be a result of SOX10's regulation of other target genes. We also then, since SOX10 isn't primarily required for turning these genes on, we don't know yet what may lie upstream of TFEC in the iridophore lineage. And finally, what we're really interested in is how these different cell fates sort out. And we're really just at the beginning of trying to understand this by looking at now more closely at our MITFA, TFEC, and other mutants. Okay, so the lab's pretty small. A couple of master's students, an undergrad, and a high school student at the moment, and then our lab mascot here. So I left myself just a little time for questions, but I'm happy to address those. Thanks.
So in the TFEC mutants, do they have increase in melanocytes in the way that MITF has an increase
we
in
just really
iridophores?
haven't looked at that yet. Yeah,
Because it
we'd
kind
like
of
to
looked
look.
like a little bit,
Yeah,
actually,
it's kind of
from
an
the
obvious
pictures.
question to
Yeah.
check out. But.
And then the other question I had is, if you, in the colorless mutant, if you don't have that lineage developing, which cells are expressing
The LTK?
yes,
and all-
LTK.
Well, those cells will eventually die off. Yeah, they just sort of hang out there not knowing what to do, but then they will eventually die. And I'll have to talk to Rob and Tom after.
Thanks, Jim.
I-
You're
we
the
gotta go.
organizer.
Yeah,
Okay,
sorry.
fair enough.